And we did talk about roots last week a little bit, but let me go back and kind of fill in the gaps and give you a little bit of a refresher. Let's see, let's start here where we looked at the comparison between dicot and monocot roots. I'm zooming into the angiosperm group of plants, the flowering plants, and looking at when you look at a flowering plant, one of the first things you ask if you're trying to figure out what it is it a monocot or a dicot? And the reason that we have them separated like that is there are definite differences, and we'll be doing this now through the rest of the semester, pointing out differences between monocot and dicot. And so when you're looking for differences, you zoom into the vascular tissue. How is the vascular tissue arranged with respect to the rest of the tissue? Usually in cross-section is how we present it. And so you can see that the vascular tissue is decidedly different in arrangement depending on dicot versus monocot. So with the dicot idea, you're looking at the central in the root. And the dicot idea is, I always think about the xylem having two parts to it, like a plus sign or cross sign. And then the phloem is in the corners of the plus sign. And then the rest of the root tissue, the bulk of it is mostly parenchyma tissue. They call it the cortex region. But the cortex region is just a bunch of parenchyma cells. And they're very alive and active and doing a lot of storage of starch and other molecules. And then we zoomed in to the monocot as a comparison and looked how that root, vascular tissue is also at the center of the root, but it's a ring of tissue. And so the ring of tissue, here's the drawing up top, where the xylem, and then the phloem just to the outside of that. And then when you look at the actual picture down low, remember with xylem, with the angiosperms, they have two types of xylem. They have vessels and tracheae. And so that's why you're seeing kind of these really big cells. Those are the vessels. And then the smaller xylem are the tracheae. But they're all clustered together in a ring. Okay. And then the phloem is just to the outside of that, more delicate looking compared to the xylem. So we looked at that under the, and this is what you would see under the microscope. So hopefully you got a good peek at that in lab with the dicot root. And then we looked at the endodermis, which is the ring of cells that can separate the vascular tissue from the cortex region. So those are the reddish cells forming a ring. And then just to the inside of that are the pericycle cells that form another ring. And those activate fairly frequently because they have to make those lateral roots out into the soil. And then this image, you can see all kinds of purple blobs in the parenchyma cells. Those are amyloplasts storing lots of lovely starch compared to the monocot root. The other thing to notice too about the vascular tissue in roots, it tends to be very central to the cross section. Not, you don't see, go back to the monocot, that make a little bit more sense. So you don't see this ring of tissue, the xylem, you don't see it way toward the frame. You see it more toward the center of this section of the root. And that kind of makes sense because, you know, roots are buried in the soil, so they have a lot of support around them with the soil packed up on them and around them. And so they can put their xylem right in the middle, and it's nicely protected from all the creepy crawly things that are in the soil with the roots. And so it's a nice strategy to keep that very precious vascular tissue kind of central, tucked away, have lots of other cells kind of buffering the vascular tissue from the soil environment where something could come along and start chewing away on the vascular tissue. What are those things coming out? Yep, lateral roots. We got the lateral roots coming out. Okay, what else can we see about roots? Roots are pretty amazing. When you start zooming into the root systems, remember they can vary, the taproot versus fibrous root, but you have kind of gradations between those two really extremes. And typically with most plants that you are familiar with, and if you're out hiking around in the woods, there's more root biomass than there is shoot biomass for most plants. There's actually more underground. Think about the iceberg idea. That's a very good analogy where what you see floating above the water is just a little bit. And then think about all the mass underneath. And so for most plants, that is true. You have more root biomass. And so it's kind of sad that we don't get to see it on a daily basis. It's all hidden away in the soil. There's a whole lot going on there. And when they zoom into the soil root environment, they start talking about the rhizosphere. So I need you to know that term. The rhizosphere, this is the scan of the front of a book. So they wrote a whole book, and there's many books just written about the rhizosphere. And really, when you look up the definition of the rhizosphere, they usually define it as that part of the soil that's influenced by the roots. And then they usually say it's about a few millimeters. And so I like the first half of the definition. That part of the soil that's influenced by the roots is the rhizosphere. And so it's like very, very dynamic area of the soil system. If you were to sample the soil right near the roots compared to soil very far away from any roots, you would see a huge difference on what's living in that soil system. And so microbes in particular love to hang out where there's a nice root system because it gives them all kinds of yummy nutrients. We say roots are very leaky, and so they're constantly rooting out different chemicals, all kinds of chemicals that microbes need for their nutrition. And in return, we see that the plants can actually selectively recruit who they want around them, and they actually will form partnerships with different microbes in order to kind of create a team effort of staying alive, getting their needs met, getting protection. And so it's a very, very dynamic area. And it wasn't until maybe the last decade or so, maybe 15 years, that we really started to appreciate how much the plant can play a role in who's around them. And so it's really kind of fun to see this dynamic that you have these microbes and these plants kind of talking to each other, chemically communicating with each other, and forming these partnerships. So with the rhizosphere, we also have to think about, okay, so the roots are in the soil. And of course, one of their huge jobs is to take up water and the minerals that are dissolved in that water. And so in order to do that, I want to start introducing the idea of this process, transpiration. So transpiration, when you think about that, you're thinking about water movement through the plant body. And it's unidirectional. We say it starts in the roots and ends in the leaves, and it's one way. You can't reverse that. And so when you think about water movement through the xylem, transpiration is the fancy term for that. But they have to get it, from the soil. The typical roots buried in the soil, they have to take up that water and the dissolved minerals primarily through the root hairs, those very precious root hairs. And so that is a very dynamic process. There's a lot of chemistry that goes into how the water gets taken up and which minerals are available. Soil systems vary tremendously in what minerals are highly available, what minerals are very unavailable to the plant. And so if you take a soils class, which you're a botany major, you need to do, you'll learn that soil is not the same. You can't just say, oh yeah, it's soil. It's all kinds of different types of soil, lots of things going on. So we say it's this unidirectional flow. Water comes from the soil into the root. Then that water has to get to the xylem in the root. And then once it gets into the xylem, that's like the internal highway system, then it can just course right through the plant body, get to where it's needed. And then actually most of the water that the plant takes up, most of it goes out through its leaves, through the stomata. It's kind of interesting how much they actually move out of their body and compared to how much they retain. So this process is important. You have to have that constant, positive flow of water coming into the plant in order to get their minerals. So that's one of the huge reasons why they keep wanting to take the water up, is because in that water are minerals that they need. They can't just make minerals. They have to get that from the soil environment. So when they've studied this process, they've named it the cohesion tension theory. And I go into more detail in this in Plant Phys, but in order for water to move through the plant body, the stomata must be open, period. If the stomata aren't open, you don't get water movement through the xylem. Not all the way up to the leaves anyways, okay? So at night here, during the summer when our plants are growing actively, when nighttime comes, our C3 plants in this area, they just close down their stomata. They take a break. So at night, they're not really doing transpiration. They save that for the day. And they also don't do photosynthesis during the night. Obviously, they don't do the light reactions during the night. Some people think they still might make sugar during the night, but they don't, because those enzymes needed to make sugar are light activated. Many of them are. All right, so water movement. Gotta have the stomata open. Why? Why is that so important? Because as soon as the stomata open, it immediately puts a tension force on the water in the xylem. And so tension is negative pressure. Okay, so it's not positive pressure. Think about sucking, soda pop through a straw. That's a tension. You're putting tension on the pop molecules or soda molecules. Which all do we say, soda or pop? Pop, soda? I guess I say pop. I know there's differences for that. So I'll do both, pop and soda pop. And so if you kind of use that analogy of drinking pop or soda through a straw, that's a good thing to think of. And so the stomata, as soon as they open up, it creates a tension force. So literally, when you look at what's happening, physics of what's happening, water is getting sucked up the plant through the xylem. And so if the stomata aren't open, you don't have that tension force. All of the tension just, as soon as you close down the stomata, the tension just dissipates and the water just kind of sits there in the xylem. It doesn't move. Stomata open, you get the suction force again, that tension, and the water starts to course through the whole body. So what's happening is stomata open, and as soon as that happens, water starts to go in the leaf. You have water oozing out of the xylem, turning into the gaseous phase, and exiting out of the stomata. So water vapor, we're talking about gaseous water now, leaving out through the stomata. So as you start doing that, that's what's actually creating a tension gradient. You have water molecules, remember now, water loves to cling to other water molecules. That's the cohesion idea. Water is very cohesive. They like to be with other water molecules. And then, so that's the cohesion part. And then the tension idea is that as the water molecules are shifting from liquid to gas, and then they're leaving or exiting the stomata, you get this pull on the liquid water molecules. And as the transpiration process keeps going, you tend to get stronger and stronger tension through the day. And so you get this nice water movement. Essentially, it's getting sucked up the plant. And you can actually see a gradient of tension. Really good, strong tension up in the leaves. And then if you were to measure the roots, you would see there's tension there, but it's less. So you get what's called a tension gradient, which you should probably write down on this slide or on your notes to go with this, because that's really what's allowing the movement. You got to have a gradient. You have to have a difference in tension from the leaf down to the root. If you don't have a gradient, then nothing's going to happen, right? So you have this tension gradient, really strong tension in the leaf, less tension in the roots. And as long as that tension gradient is maintained, you'll have the water getting sucked up through the plant. So it's actually pretty slick mechanism, because remember, is xylem living or dead? It's dead. So there's nothing in there, just like a straw, like drinking, soda pop through a straw. It's just this hollow tube system, all the way joined from the roots all the way through to the veins of the leaf. And so it's a very efficient movement of water. There's no cytoplasm in the way, no organelles that the water molecules bump into. It's just get it to the xylem, and it's on this very quick highway system. It's just going to go. And so we'll come back to this idea of transpiration as we get into stem and leaves. I'll keep revisiting it because this is a pretty important concept to think about. It has a lot of impact on how you manage your plants and how they end up growing or not growing. Are there questions right now on transpiration? Okay, well, I'll, like I said, touch base on that again, but let's finish off the root idea. When you're thinking about roots, they also can do more than just take up water and get that going through the plant body. Certainly, they can be used to clone themselves. So often in the spring, you'll see new stem and leaves sprouting up from under, roots or underground stems as well. So you can have cloning going on. Vegetative means you're basically cloning. Asexual again, cloning. You're cloning the plant through the root systems, growing fresh stem and leaves in the spring, for example. Certainly, storage is a huge component of roots as well. Fall with our plants, they're very, very busy bringing down all the nutrients out of the leaves, getting it stored into the roots, and then they'll let their leaves drop before winter. Certainly, access to nutrition. We'll talk about some parasitic roots that are busy parasitizing other plants. And then some plants, when you think about where they live, along the coastal shores, for example, along lakes and rivers, you have, especially if you've gone, last time I was in Florida, I went to the mangrove swamps and got to see mangroves. They had boardwalks. You could just walk right amongst all those amazing trees. And you can see all the snorkel roots. And so snorkel roots are just these roots that tend to stick up out of the muck. And they are, if you look at that tissue, they're very airy tissue. And so it's really helping with airflow into the roots. Because remember, roots are living cells. They need to breathe. They have to have oxygen coming in, just like, we have to have oxygen to stay alive. And so they gave it a special name. They call it parenchyma tissue. A-E-R-E is referring to air. And so it's very nicely adapted to when low tide is occurring, those roots will become above the water, and then they'll just be able to really take in all that lovely air and oxygen. And then they often say that when high tide comes, they hold their breath because they're submerged again, and it's much tougher to get the oxygen out of the water. Much easier to do it. Movement. Certainly, there's when a seed is germinating, that's probably the best time you can start thinking or witnessing this movement, where you'll see the roots starting to grow, and they can actually kind of twist and coil and kind of corkscrew their way into the soil. Some will kind of contract and relax, and it causes them to get really deeper and deeper into the soil. So they have strategies to really anchor them through these movements. They're not super quick. You have to be patient to watch this or speed up the video. So they gave them the name contractile roots because many of them are doing that, or corkscrew roots. And there's some nice video too of roots, or seeds, I should say, seeds that have modified appendages coming off, and it looks like a little corkscrew. And then when they land on the soil, they get wet and dry, and that shifting from wet to dry causes the corkscrew mechanism to, so they they drill into the soil. It's pretty amazing. If I don't have it on the slide set, I'll post it. It's really cool to look at. So nutrient, here's some visuals now of what we were just talking about. Certainly, I think if you thought about it for a little bit, you come up with some of these as well. Carrots and parsnip, you looked at parsnip in lab, sugar beets, things like that. Don't ignore the bananas, but everything else on there is cassava and taro, which we don't grow around here. That is found in tropical, some tropical, but cassava in particular is hugely important crop. It feeds millions of people. You can see they're quite large. The cassava are the lighter ones. So certainly nutrient storage. This is a manroot plant, and they're showing the huge the roots that basically has been really enlarged for water storage more than anything else. And so, um, very, very interesting. Here are the ideas of the snorkel roots. Oh, another fancy term for them are pneumatophores. So, pneuma, breathing. For anytime you see PHOR on the end of a science word, it means to hold or to bear. And so we use that for root all the time in different words in science. So parenchyma tissue, snorkel roots. So here you're looking at the shore with mangroves, and then there's a lower figure, they're showing a close-up of the snorkel roots. So really nice adaptation for these trees to be able to get enough oxygen through their day, through a 24-hour period. There's another good example of low tide where you can see all the snorkel roots. I believe you also had a living Elodea plant to look at. Some people say Elodea. I say Elodea. This is an aquatic plant. You can find it around here. It was kind of hard to go find it right now, so we went and bought one. But you can find them around here all over the place. And if you notice, when you looked at it at lab, not much xylem, really. When you look at the prepared slides that we had, it's kind of overkill to build a big xylem tissue because they're always in water. So they can just kind of soak it up. They don't really need to have a designated xylem. They'll have foam, but you really don't see xylem. So they're just basically absorbing. They don't need to worry too much about transporting water like most plants. They're pretty much totally submerged or mostly submerged. Now, these aren't mangrove trees. These are actually corn plants, and those are actually roots that come off the base of the stem, but they're above ground, or good, most of it's above ground. But if you think of it, if you are kind of think about architecture, having poplars like that at the base of the stem, if you ever walk through cornfield, they can get quite tall. And so it kind of makes sense that they have that little extra base like that to really help stabilize them so they're not getting knocked around too much with wind or something coming along and bumping into them. And so those prop roots are very functional. They're cool to look at. And then on the right here, you see orchids. You looked at orchid roots too in lab, where you were noticing that orchid roots tend to have multiple layers of epidermal layers on their roots to protect and help hold in the water better. And then they often are green. That doesn't really show up too much there, but green roots for orchids makes sense. Most orchids hang out on tree branches, not the ones up here, but the ones in the tropics. They like to hang out and grow on tree branches without harming the tree. They're not doing any damage. So their roots are totally exposed and they tend to green up and actually do some photosynthesis. Here's a really dramatic example of prop roots. Okay, so this is a bigger tree. This is called a screw pine tree, and so they grow much bigger than corn. So you usually see this kind of system in areas of the world where the soil isn't that great. The deeper you go, they're kind of like the roots are kind of limited to the upper portion of the soil system, and either they can't go deep because of bedrock, or there's not any nutrition to search for down there, so they don't. And if that's the case, if you have really shallow root systems, yeah, you need a little extra help with making sure that your above ground stuff doesn't topple over and you're kind of secure in place. Here you see another example of an orchid. This is the vanilla orchid, and it's a vining plant in the tropics. It likes to vine around trees. And that's, again, you can kind of see how parts of the roots dangling there are greenish. And then orchids you buy at stores, they tend to, I don't know if you can see, but there's usually the healthier the orchids are, the more they'll start to send out these aerial roots like that. And those are nicely green. You can see that's a very healthy. That's not my orchid. A friend of mine sent this. So, but there's a close up of really big root system. So this orchid is very happy. Any of you who live down south know what kind of tree this is? Oh, it's right there. Banyan. I didn't know. Never mind. It's banyan. So yeah, the first time I saw these, I thought, well, what those are the roots that start with branch tissue. So it's branch tissue, and those are adventitious roots that start to just grow down and then bury into the soil. And this is a very old section of banyan trees. And you can see quite extensive tree roots. It's pretty amazing. And so some cities, really play up the banyan tree. They landscape around their towns and cities around down south for a bit. So very, very impressive looking. Some roots are coming off like the whole length of the stem. Here, this is poison ivy. And poison ivy likes to cling to other things, whether it be a brick wall or tree trunks nearby. And so adventitious roots can be big or small. And it's not, we've been talking a lot about adventitious roots with stem cuttings, but just be aware, these are just adventitious roots coming off the whole length of the stem, not because of a stem cutting idea. And again, it's just helping them secure themselves to whatever it is they're pressing against. Brick wall in this case with the Boston ivy. And then if prop roots get a little bit more massive, then they start saying buttress. They'll say, oh, look at those buttress roots. And so those are like prop roots on steroids. That's a fig tree. Again, fig trees can get quite tall. And fig trees are in the tropics. Tropical soil is pretty poor. It's pretty shallow in terms of the quality of soil is pretty shallow. And so it really does help them with getting stabilized because their root systems that are in the soil are not very deep. Really massive buttress roots. So, trees in particular can be master architects in this regard. They can really develop their body to deal with their environment, whether it be shallow soil, shallow roots because of that, or really windy conditions. And so they can adapt really well to their environment. This is the idea of the contractile roots, where it's basically just a wet, dry action. As the tissue gets wet, it'll start to swell. As it dries, it shrinks. So you'll get this kind of kneading action of the roots. And that, if you do that in the soil long enough, it keeps you going down, down, down. So that's what this artist is trying to show. And so dandelions are really good at that. If you've ever been given the job by your parents to go dig the dandelions out of your lawn, their tap roots are deep and really hard to pull out. And actually, in my opinion, you shouldn't pull them out. Dandelions are awesome plants. So, all right, mistletoe. So these are actually parasitic plants, which is kind of funny that we make such a big deal around the holiday with kissing under the mistletoe, and it's a parasite. And not only that, it's like one of the worst parasites, in my opinion, because it's a hemi parasite, technically. So hemi, if you put HEMI in front of the word parasite, it's a hemi parasite, which just means it's a half parasite, essentially. It can do photosynthesis on its own if it wants to. It just doesn't want to, so to speak. It's easier to be parasitic. And so this is a group of trees that are heavily infested with mistletoe plants. So, by the way, in order to get those to the store to sell out to people to hang mistletoe around the holidays, so these mistletoe in the United States are in the southern regions, the United States, and they're usually in dense forests. So they can't like really go to the trees and climb them. That's not very efficient. So they actually shoot them down. They go mistletoe hunting. I always wondered how they get mistletoe. And I was listening to a podcast, and they were interviewing father son, and that was their job. That was their livelihood. They went and hunted mistletoe, and they got way into it. They're like, yeah, what kind of gun, rifle. They used a rifle, and what kind of bullets, and where to shoot, so that you don't damage. You don't want to obliterate, the clumps. You want to keep them big enough so that you can sell them in more nice looking clumps. And so, yeah, there's a real art to it. So that is much more efficient than trying to drag a ladder through a dense forest. Yes. Not really, because what they do, I have a video I'll post in the reading and video section. So the other thing is, how do they get there, right? I mean, it's a totally different plant than the plant they're growing on. It's like a parasite. And what happens is the birds come along and eat the fruit of the mistletoe. And these birds are, hanging out on tree branches, going from mistletoe to mistletoe, eating all the fruits. But then when they poop out the seeds, it's really gooey and slimy, and it starts sticking to the tree branch. And not only that, but it sticks to the bird's butt. And so the bird, there's a really good video, I'll post it. The bird like wipes his butt along the branch because he's like, yeah, I got a dangling thing on my butt. And so they kind of hop along and rub their butt, and it rubs the seed, and it's so gooey, and so it sticks right to the branch. And then they show videography, they speed up the videography, and you see the radical root coming out and just plugging right into the branch. It's like genius. Like that is such a cool adaptation. So they, with the help of that very special bird, they call it the mistletoe bird. That's how it spreads amongst the tree branches. So pretty slick mechanism. I'll post that video because it's really funny. It's a short little video. So mistletoe is all over the world, hanging out in branches like that, and they like to plug in their roots to tree branches and start sucking away all the nutrients, primarily through the vascular tissue, the water and the sugars from the xylem and phloem. This we have around here. So this is a Monotropa plant. That's the genus name. And it's given many different common names. Ghost plant, ghost pipe is one. And doesn't that look more like a fungus? But it's actually an angiosperm parasitic plant. And so you can see it's not really green at all. It doesn't do any photosynthesis. So this one's totally dependent on a nearby plant. It can't do photosynthesis like the mistletoe can. And so if you see this popping up, we have this, and there's a patch that I found not too far from here, actually. But they're busy underground with their roots. What they do is they find a mycorrhizal fungus, which we'll talk about in this class. They team up with the mycorrhizal fungus. Mycorrhizal fungus plugs into another tree, and so it's a bridge. Like the plant is using the mycorrhizal fungus for some of its, give me some of those nutrients from that tree you're hanging out with. So it's very parasitic. No, I mean, like mistletoe can hang out on different plants depending on where it's like. There's eucalyptus trees in Australia that have mistletoe, but down south we don't really do a ton of eucalyptus over here. So there's other plants that it hangs out here. With these plants, they're usually, I find them in a mixed hardwood forest usually, and they think that they're actually using the mycorrhizal fungus to get nutrients from a variety of plants nearby. So it's not like this isn't too picky. Like I'm only going to hang out with red pine trees. It's pretty broad. Mhm. Mm, okay, so they might be a little picky about the fungi. Yeah, yeah,
So how many know like scrub?
yeah. So, one of my grad students, Carrie, she also did, she was looking at orchids, not this one, but she was looking at orchids and the fungi those orchids hang out with. And we were noticing a trend, like this orchid seemed no matter where it was. So there's a patch of ram's head lady slipper orchids that are going extinct around the world. We have a huge patch of them in the Isle Royal. What am I thinking of? You know, along in Munising, where all the sand dunes are. And they estimate over 2 million of those orchids there, where everywhere else it's starting to go extinct. So Dr. Robertus' grad students and then one of my grad students were like, why? And so Dr. Robertus's grad students were looking at some of the above ground stuff, and I said, let's look below ground. So my student was looking at what kind of fungi were in those areas, and we were finding a couple of different species of fungi that seemed to really like hanging out with the ram's head. So you get those types of dynamics too, and you know that often because it's all underground, it's really hard to study. It's so, but I always like to interact with Dr. Robertus' students. So don't forget what's happening underground because a lot of that underground dynamic is influencing what they're seeing above them. This is a not so nice plant. It sure looks pretty though, right? It's nice and green with the lovely red flowers. So this is a striga plant that is growing at the base of a corn stalk. And striga, they actually named a plant hormone after this. They call it strigolactones. And strigolactones are a relatively new hormone group. And the reason they named it after striga or striga, that parasitic plant, is that when the plant releases strigolactones, what it's trying to do is attract mycorrhizal fungi. However, this plant cues in on it. It's a sneaky little plant. And so it's like, oh, I smell strigolactones near me, so I'm going to germinate and grow and parasitize. Where the plant wasn't trying to get that to happen. The plant was trying to call out to mycorrhizal fungi. And so this strigolactone is kind of eavesdropping and picking up on that hormone. And then that stimulates it to grow. And then here, this bottom portion of this image on the right, that's the root system of the parasite, and it's docking into the corn stalk and it's starting to parasitize. So this one's a huge problem in commercial crop fields because the seed bank, the seeds there, they can just persist and persist. And then if the strigolactone hormone's getting released, they start to pop up really crazy. So the fancy name for their parasitic root is called a haustoria. You don't need to know that, but you might see that word in other science classes. It's a common way that we talk about roots. But it's done a lot of damage commercially to yield of the crop you're trying to grow. In that case, that was a cornfield. And so they're really looking hard at how do we get rid of striga? How do we control it? How do we obliterate it? And they're trying genetic engineering. They're trying to spray the strigolactone early before they plant their crop, get the striga to grow, then they kill it, and then they put the crop in. So it's a lot of extra work that is causing, extra issues with paying for labor. It's definitely so unfortunately, it's just really good at eavesdropping. When the plant starts releasing the strigolactone, it's very good at getting that cue. Okay, so root systems, massive. All right, very dynamic. This is one that blew over in a windstorm. That's my brother. You can see he's over 6 feet. So, and that's just part of it. Uprooted itself. So root systems are very big in general, bigger than what you see above ground. Very dynamic area of interface between the root and the soil and all the organisms in the soil. Wow, it's already 12:45. All right, so I'm gonna stop there and, let me just go over a few things on the exam 1. So let's see. Um, huh. So I did the stats and I ended up adding 5